Well, thank you very much. Thank you for the introduction and the invitation to speak to you this evening. I'll just get my technology working, and I think I'm ready to go. So eventually, I'm going to talk about the subject of my title. But before I get there, I would like to give you a little background, and first of all, take you back to 1973, when I decided to do a PhD. And I asked my PhD supervisor what I should do as the subject of my research, and he said, well, that's for you to decide, but what do you think is the most important question in biology? That's what you should do for your research. He said, you're going to have to devote yourself to this project, and so you don't want to be messing around on something peripheral. What do you think is the most important thing? And so I decided at that time that for me, the most important question in biology concerned genetic regulation. So these are three figures who will feature at various points in my talk, but I'd first of all like to start with genetic regulation. And for those of you that don't think about this sort of subject every day, just like to start off and saying, well, what do I mean by genetic regulation? And I think this can be best explained in terms of a very simple experiment that involves taking a plant. I'm professor of botany, I work with plants. And in this simple experiment, you just chop up your plant and take individual cells from different parts of the plant. And if you then culture these cells under the right conditions, you can regenerate a whole plant. So what this tells you is that the cells in the shoot have got all of the genes that you need to make a whole plant. The cells in the root have got all of the genes that you need to make a whole plant. So from Francis Crick's central dogma, he tells us that DNA makes RNA, makes protein. We know that the proteins in a root cell are different from those in a shoot cell, and so on and so forth. Same applies in animals, feet and brain cells, and so on and so forth. And so what we can infer from that is that some of the genes in a shoot cell are switched on and switched off, and other genes in the root cell are switched on and switched off. And so genetic regulation is all about understanding these switching on and switching off processes. And I took a sort of reductionist view at the time. I thought that if one understood the switching on and switching off of genes, that this would help you understand organisms as a whole, and then beyond the organism, it would help you understand populations of organisms and so on and so forth. So you could argue that I was a naive reductionist at the time, and maybe I still am. But it seemed to me that it was an important question, and so that's what I set out to tackle in my research. Now, at various times, so this just shows you the situation that we had in 1973 when I started all of this. So we knew about genetic regulation in bacteria from the wonderful work of Jacques Monod and François Jacob, for which they got the Nobel Prize. And so their work told us that if you have a gene, whether or not that gene was switched on or off was influenced by proteins that bind to the region of DNA adjacent to the gene. So it was a very simple sort of process, and those proteins could be influenced in turn by molecules in the environment. So this gave us a connection, an understanding of how it was the environment could influence the switching on and switching off of genes. In more complex organisms, I hesitate to use the word higher organisms, since that is a value judgment, and who's to say what is higher and what is lower. But in more complex organisms, plants and animals, there were lots of theories, and this was one that influenced me hugely, some of which were remarkably prescient, but there were very few data at the time with which to base our understanding of genetic regulation. So that's what I started off doing in my PhD, initially in Edinburgh, and then I did postdoctoral study, as you heard, in Montreal and Athens, Georgia, where I have to say I encountered only very minimal racial discrimination. I had people come along to me and tell me that they really had almost forgotten the War of Independence, and so they were prepared to tolerate me as an Englishman in the South. But anyway, and I had a wonderful time there. And then I came back to the Plant Breeding Institute in Trumpington in Cambridge. It's where Waitrose supermarket is right now. And if you go to checkout number 4, that's where my bench was at the time. So you'll understand if I shed a little tear every time I go and buy my cornflakes from Waitrose supermarket. But anyway, that's where I was. And by this time, I'd started working with viruses. And I'd started working with viruses not because they cause disease, although I was interested in that, but I started working with them because they're basically tools with which you could study genetic regulation. So viruses, for those of you who are not biologists, they're basically little packets of genes. And they're very easy to get hold of. They're very easy to work with. Their genes are regulated. And viruses also regulate genes in the host plant. So here is a virus disease. And what is happening is a gene is being switched off that is responsible for the green pigment in the leaves of the plant. So the leaves actually look a rather wonderful sort of yellow color. So viruses are wonderful tools with which to study genetic regulation. And so that's what I was doing. And we had some successes at that time, and the successes were sufficient to get me another job, a job this time at the Sainsbury laboratory that was in Norwich. And it was basically an experiment set up by David Sainsbury, who wanted to support a laboratory in a topic that he thought he could, the resources available to him, substantial as you know, would be sufficient to make a difference in a field. And I hope that they did make a difference. And so while I was there studying genetic regulation, I also started working with the newly developed techniques of genetic modification. And we were using this as an experimental tool. So this is basically a primer in genetic modification of plants. And the key molecule, as always, is DNA. And this was a methodology developed by a friend and colleague of mine, Michael Bevan, and others. And what Michael and his other colleagues were able to do was to develop a method whereby you could take some DNA, for example, from a virus, and you could transfer it firstly into a bacterium, and then the bacterium would take that DNA and they would insert it into the chromosomes of the plant, so that that DNA was then inherited along with all of the other genes. It becomes an integral part of the chromosomes of the plant. Now, so David Sainsbury said to us when he set up the laboratory, he said, well, you know, what I want you to do is basic research. But, he said, if you ever see the opportunity to do something useful, then I want you to take advantage of that opportunity. And what we could see was, having acquired the ability to work with the genes of viruses and having got this genetic modification technology, this developed by Michael Bevan and his colleagues, there was an opportunity to try and develop virus-resistant, disease-resistant plants. And the idea was based on a concept known as parasite-derived resistance. The concept's very simple. You take a gene out of a parasite, and you use technology like this to insert it into the genome of a host organism. And what you might expect, for various reasons, is that having got a new gene into the chromosome of the organism, of the host organism, a gene derived from a parasite, that this would somehow immunize the host organism against the virus disease. And so what you would expect is that the organisms you had generated carrying the gene from the parasite, that these organisms would be resistant against the disease. So that's what we tried. So this is a plant virus, doesn't matter what it was, and we took a gene out of it, and we transferred it into plants, and then we asked, are those plants resistant against the virus disease? And the answer is they were, so we were very happy. But there was something rather strange about the results that we got. So this is where Thomas Kuhn comes in, because Thomas Kuhn tells us that scientists mostly are doing what he calls normal science. You're basically validating what you think you know already, and what you're always on the lookout for is a result that doesn't fit the existing paradigm. And so here it seemed as if we'd got a result that didn't fit the existing paradigm. So how did this result not fit the existing paradigm? Well, when we looked at our different transgenic plants, we got several different transgenic plants, all carrying the same gene. And some of them were resistant against the virus disease, so no symptoms on the leaves. Some of them were fully susceptible. So here, this mosaic is the symptom of the virus disease. But what was rather strange was that in these plants that were fully susceptible to the virus disease, the gene that we had put into the plant was switched on. It was very active. In these plants that were fully immune to the virus disease, and they were impossible to infect with the virus. We could take concentrated preparations of virus, inoculate it to the plants, and they never got infected. So these were highly resistant plants, but the gene that we had put in was apparently switched off. Remembering that DNA makes RNA, makes protein, these genes apparently were not making any RNA at all. And that just didn't make sense. How is it that a gene that is apparently switched off can have an effect like this? It can stop a virus, whereas the same gene, when it's switched on, has no effect at all. Well, I'm embarrassed to say it took us a couple of years to work out what was going on, and I expect that most of you in the room here have worked it out already. It was actually very simple. So what was happening was that we had got our virus DNA, and it had triggered something that we called silencing. And the silencing was switching off the RNA from the transgene. And because the RNA from the transgene has got identity to the virus, what it was also doing was switching off the viral RNA. So this process, this silencing process, was explaining how it was that our transgenic plants could be resistant against the virus. So we then beavered away for a while, and we tried to work out what was going on, and we actually made quite a lot of progress. What we also found was that actually, there's nothing new under the sun. So we thought we were being, a bit smart here by engineering plants to be resistant against a disease. But it turns out that this is the same mechanism that most plants use anyway to protect themselves against virus diseases. So when a plant is infected with a virus, it's not fully susceptible. It's still able to stop the virus, and it's able to do that because it's doing this silencing process. So this just shows you a plant infected with a virus, and this shows you a plant that can't do RNA silencing. And as you can see, it's really super susceptible to the virus disease. So this underpins the idea that was mentioned in the introduction, and I'm absolutely convinced that it's right, which is that if you've got aspirations in biotechnology, it's much better to say, well, what does the organism do anyway? And let's just try and tweak it a bit, rather than trying to introduce a new synthetic biology into an organism, a person or a plant or whatever. Just have a look at and try and understand what the organism does anyway, and just tweak it a little bit. And that basically was what we were doing in our plants. So this gave us, the RNA silencing and the understanding of the virus resistance. And what I'd like to do, I'll just show you a little cartoon here that illustrates what's going on. And before I show you the cartoon, I just want to remind you about Watson-Crick base pairing. So you probably all know that DNA is made of A and C and G and T, and the A's pair with T and the G's pair with C. And so I hope that's not too technical, so that's a fundamental principle that underpins what is going on here. And I'm going to show you inside a cell. So we're going to be looking inside a cell, and in the background you'll see a nucleus, and in the foreground you'll see a viral RNA molecule. Remember that we're looking at a virus defense mechanism here. So here it goes, and so here's the viral RNA, and there's a protein in the cell that can take that viral RNA molecule, it's RNA from the virus, and copy it into a double-stranded form. So it's copying the A's and G's and so on and so forth. And then there's an enzyme that comes along and chops that double-stranded RNA into small little fragments. And my lab, I'm proud to say, were the first lab to see these little small fragments of RNA. And then what happens is these RNA molecules get bound by another big protein in the cell, and they lose one of their two strands. You can see it being lost here. So now you've got a big protein which is attached to this little small RNA molecule, and this can roam around in the cell, and if it sees another RNA molecule, through Watson-Crick based pairing, it can bind to it, and this big protein that's bound to the RNA, it can chop up RNA. There it goes, it's chopped it up. And so, of course, that viral RNA is no good anymore because it's chopped into two, and it's even less good because there are some munchkins that come along and hoover up the spare fragments of RNA in the cell. Just think about it for a while. This is a defense system, and it's absolutely brilliant because what it means is that I think we can turn the lights up now, thanks. What you've got is the specificity of this defense system is taken from the invader. You know, if you want to defend against an invader, you always want to make sure that your defense system is hitting the invader and not anything else. So if you get the identity of the invader from the invader itself, then you've got the ultimate specificity, and that's what you've got in this rather wonderful virus defense mechanism in plants. I just want to make a couple of points here. So I have to say, so we set out to make virus-resistant plants, but we didn't expect that we would be doing anything with RNA. So what we came on, and the discovery of the little small RNA molecules, was basically serendipity. It was, we got lucky, basically. And we're not alone. I mean, the whole history of science is littered with stories of people who got lucky. So here's Alexander Fleming. This is Helicobacter pylori, which if you're unfortunate enough to suffer from stomach ulcers is probably the causal agent. And it was discovered by a couple of Australians, one of whom is called Marshall, almost by accident. And this is another one. I got these examples from this paper here. This is the use of lithium as a medication for people unfortunate enough to have bipolar disorder, and this was an accidental discovery. So serendipity in science is hugely important, and so having a benefactor, a funder of science that recognizes this and supports an environment where you can get lucky, I think is hugely important, a lesson perhaps for the UKRI and the government supporters of research. So that was one point I wanted to make about the importance of serendipity and accidental discoveries in science. Another one, this is a more general point, which is that, so these little small RNAs that we found and the whole process of RNA silencing turns out not to be specific to plants. It's also active in animal cells as well, and even some unicellular organisms as well. So it's a process that existed in a primitive cell before the evolutionary divergence of animals and plants. And it turns out that there are lots of these small RNA molecules, not even corresponding to viruses, but corresponding to parts of the genome. And so this led to one of my colleagues referring to this small RNA as being the dark matter of genetics. And it's quite an apposite metaphor, because like the dark matter of the physical universe, we think that these little small RNAs in cells have, let's say, a coordinating role. They sort of bring everything together, just as does dark matter in the physical universe. But I suppose the point here is that this is all stuff that came from plants. It's rather gratifying, though, that some people have picked up on this, and they came up with the idea that maybe what you could do is, if you could identify genes in people that cause disease, what you might be able to do is to design little small RNA molecules that would knock out that disease gene and protect people against the disease. And the first RNA drug got approval for clinical use in the USA earlier on this year. So it's rather pleasing to see a discovery that was based in part on work in plants actually finds application beyond plant biology in biomedicine. This isn't the first example. The whole history of biology has got many, many examples of where plant science has made some fundamental contributions to biology. So these are Gregor Mendel's peas. We can go back to Robert Hooke in the 17th century, one of the pioneers of microscopy, and he was looking at sections of cork, and he saw these little compartments in the cork, and he compared them to monastic cells. So this is the first use of the term cell in biology by Robert Hooke, looking at plant samples. Beijerinck, a Dutch scientist, discovered what he called a contagium vivum fluidum. So this was an infectious agent that could pass through a porcelain filter. It turned out to be a virus. So this was the first report of viruses in plants. So whenever I think about all of these things, I'm always reminded of an undergraduate essay that I had to write when I was a student. And the topic was, "Botany is monotony. The study of plants I leave to my aunts. Discuss." And I have to say, I think I made rather a feeble job of it at the time, and I hope that if I were ever set that essay again, using the sorts of examples that I've given you just now, I could make a better fist of it. But anyway. So this is, I'd say, some general stuff, but what about agriculture and crops and everything together? So first question, is there a place for GM crops? Well, that depends on you. My view is that there is. And I think you could forget almost everything to do with GM crops, and you could just say, well, let's just think about disease resistance. I've shown you how you make plants resistant against virus disease using GM. This is part of a trial in Norwich looking at plants that were resistant against late blight. Any of you who are gardeners growing tomatoes or potatoes, you'll be plagued with late blight quite often. So there's a GM solution to all of that. There are many other GM solutions to disease problems in crops, irrespective of the causal agent of the disease based on GM. So even forgetting the other traits, you could say, well, yes, I think there is a place for GM. The other traits that can be tackled with GM include all of these traits here: water efficiency, fertilizer uptake, nutrient quality, post-harvest loss, enhanced photosynthetic efficiency. So there are lots and lots of examples. But as you probably are all aware, there's huge resistance to the introduction of genetically modified crops. So you've got two camps. Some people who say, this is good, I think we should use it, and other people who are not so keen. I won't mention any other words, but we're all seeing what happens when you've got two camps of people who really can't see a way forwards. And so what I want to do is just, in the next few minutes, think about, how do we go from here? And my thinking about this was influenced very much by a report that I was involved in for the Royal Society, actually about 10 years ago. And so this started around the table in the Royal Society, and people were saying, well, should we make a position statement on GM? And what we thought was, well, there's no point in just making a position statement on GM because crops and agriculture are a huge topic. And so what we wanted to do was to think more holistically about science and the issues with crops and agriculture. And one of the members of the working group, a chap called Jules Pretty from Essex, came up with this term here, sustainable intensification. And I think our report was probably the first time when this term was actually used. So what we were doing was we were thinking about, how can you use science, bioscience in particular, to deal with some of the issues in crops and agriculture. And what we could see is that there were a number of issues with crops and agriculture. So I think you could put them under two headings. So one is sufficiency. Are we confident that we're making enough food on the planet for all of the people? And the other one was sustainability. So even if we can make enough food now for all of the people on the planet, are we confident that the practice that we're using is such that we'll be able to continue to do that indefinitely? Because hopefully the human race will be on the planet indefinitely. And you can have a discussion over the sufficiency topic, and I'm happy to pick up on that later. I think that sufficiency is something we can deal with, but sustainability, I think, is more of a challenge. And you could use a number of examples about sustainability. So you could talk about aquifers and water. You could talk about soils, soil erosion, soil carbon. You could talk about all of those sorts of things. You could talk about biodiversity and how biodiversity in agricultural environments is often reducing. But here's one example, and it just shows you how, let's say, some practice in agriculture is a bit wasteful. This shows you what happens when a breeder develops a disease-resistant crop. When it's introduced, it starts off being fully resistant, but within a few years, it loses the resistance. And the reason is that when you grow your crops, and particularly when you grow your crops as a monoculture, which is what we quite often do, then you place the pathogen under very strong selection pressure to overcome whatever resistance you have put into the field. And so these resistance resources are being squandered time and time again. So we've got a challenge. We've got to develop various practices that will help us deal with this sustainability component in crops and agriculture. And in the report, we came to the conclusion that the solutions to sufficiency and sustainability in agriculture have got three components to them. Solutions based on genetics, solutions based on crop management, and solutions based on chemistry. And we were particularly keen on solutions based on genetics and solutions based on crop management. Chemistry has got complications, as you're all aware. And so while we didn't want to rule out the importance of chemistry, we just thought that the potential of science to contribute to genetics and crop management was quite significant. So what do I mean by genetics? Well, in the spirit of wanting to have biotechnology accepted generally, what we're doing for the moment is not talking about GM, or at least I'm not in this lecture. I'm an advocate of it, as you can see, but what I don't want to do is to get bogged down in a discussion of GM. So let's just think about other things that biotechnology can do in the way of genetics. And I've only got, the 45 minutes this evening. I'd love to have 3 hours when I could take you through all of the fantastic advances in bioscience these days that are associated with DNA sequencing and various other advances. But what it boils down to is that even with conventional plant breeding, it is hugely more powerful now than it has been in the past. So there's a short cycle, it's faster and more precise than it ever has been. And so the potential of genetics using DNA sequencing technology, no GM, parking that for the time being, just genome sequencing and so on and so forth, gives us huge potential. We can diversify crops and agriculture a lot more easily than we couldn't even think about doing some of the things that we can do right now. So that's one thing. So another sort of set of technologies that we drew attention to in our report were crop management strategies. And I just want to talk about a couple because these are not things that you might normally think about in terms of biotechnology. So this is in Africa here, and this is an approach developed by my friend and colleague John Pickett from Rothamsted, and he called it push-pull, or companion cropping. I mean, it's a technique widely used by organic farmers and others. And the idea is that you've got a maize crop, and around the maize you grow a forage grass called Napier grass, and in between the maize you grow a legume called Desmodium. So why do you do all of this? Well, you do all of this because what happens is that the Desmodium produces a volatile compound that repels a stem borer moth that does damage to the maize. The Napier grass produces another volatile that attracts the stem borer moth, so hence the fact push-pull. The stem borer moth basically is being guided away from the maize. The Desmodium, being a legume, fixes nitrogen, so it fertilizes the soil, and there are other advantages to do with parasitic weeds and so on and so forth. So it's a wonderfully elegant technology. It's not easy to deploy, but nevertheless, it does have a number of advantages, and the figures are truly staggering. Where this has been deployed, the yields of the farms with no inputs have gone from less than 2 tons per hectare up to, in some instances, nearly 6 tons per hectare, without any fertilizer or irrigation or whatever. So truly staggering, and an illustration of how this type of approach could be useful. I want to give you another example here. So this is a picture of another colleague, Martin Wolf, who sadly died a couple of weeks ago. He was at the Plant Breeding Institute when I was there, and he was at the time an advocate of growing mixtures. So rather than growing one variety of cereal in a field, what he liked to do was to grow mixtures. Well, why would you do this? The first reason for doing this was because it basically gets away from monoculture. So it gets away from this disease susceptibility problem. So if you grow mixtures, you're making it more difficult for diseases to spread within a field. But this just shows you in 2012, on Martin's farm, one of the trials that he was doing. So you've got these plots, which are single varieties, and then these plots that are mixtures. Now, for those of you who've ever been on a farm in East Anglia, you'll probably recognize that this monoculture is not looking very good. and it's got a sort of mist all over it, and this is called blackgrass. It's a big weed problem for arable farmers, particularly in East Anglia, but I guess throughout Britain. Now, for some reason that we don't understand, in the monocultures, there was no protection against it, but in the mixtures, what we don't understand is why the mixtures were somehow protected against the blackgrass. So I think, well, what this is telling us is that there are some benefits to growing mixtures, some complications associated with growing mixtures, but there's also interesting science. What's happening in the soil? How is it that the mixture can protect the crop? So then, how do we bring all of this together in a reconciliation that at least will move us forwards? Well, I'd like to propose so on the one hand, you've got this fancy genetics, but until now, it's been deployed simply in the context of what you might call industrial agriculture. But if we tried to link it with the practice used by organic and traditional farmers, I think there's potential for huge advances that would maybe reduce the inputs and some of the environmental costs associated with agriculture. I know it won't be an easy task, but it would be made easier by the power of the biotechnology that we're developing. And who knows, once one starts looking at all of that, and if you have the different camps, the different protagonists talking to each other, then maybe what they would start thinking about is, so the biotechnologists would start understanding why the practitioners of organic don't like GM, or maybe it would work the other way around, that the practitioners of organic would start understanding what are the benefits and the advantages to GM. But without having a discussion and without having interaction, I think it'll be very difficult to make progress. So I just want to close with this, and this is from the preface of Micrographia, so the book in which Robert Hooke described cells, and it's got all of his wonderful pictures of fleas and cork and all stuff. But he says, in his preface, he says, it's the great prerogative of mankind, above all other creatures, that we're not only able to behold the works of nature or barely to sustain our lives by them, but we also have the power of considering, comparing, altering, assisting, and improving them to various uses. So I think at the moment, at least in crops and agriculture, we're not really taking full use of our prerogative. And I hope that by a bit more lateral thinking and dialogue between people engaged in different sorts of agriculture, that we can do that. So with that, I'll close, and I'll be very happy to take questions.